So we will be moving after the break into our breakout sessions. And so we have 4 different groups, and just in a brief summary of what each context of use is to orient you and not take up an entire page. So there's the patient stratification, and Klaus Romero will be facilitating in that room, in the Wisconsin room. And then the next is looking at de-risking class toxicity, and that will be in this ballroom, and Katrina will be facilitating that meeting. And then for retrospective toxicity analysis, we'll be in the Cordell room, and I'll be facilitating there. And finally, looking at immune-mediated DILI, Graham Marsh will be facilitating that in the Rosedale room. And so here's the list of where everyone wound up. Most everyone, your first choice is where you are assigned. A few of you, we needed to balance out a little bit, and so a few people were moved, but not very many. After we go through, I think the next slide, we can go ahead and I'll come back to this slide. Oh, no, excuse me. So the first piece I just wanted to say is, as we move into the breakouts, I think that Tina, your talk is a wonderful place to bring in mind as we're moving into the breakouts. And then there are the 2 handouts, one from Tina and one from us, looking at different considerations as one is looking at putting together a qualification project. And so within each room, our goal there is to look at the broad context of use, put that into a more refined and in the format that's acceptable, and then look at what the analytical considerations should be, as well as the biological performance considerations. And within that, we'll come back after lunch and summarize in this room. And I believe with that, we have about a 10-15 minute break. Or are we going straight in? Okay. Well, why don't we, let's see, right now we're at 5. So yeah, so let's everyone, let's meet in your respective room at 10:15 and give everyone a brief bio break and time to grab another drink. All right, thank you everyone for joining us here in this last half of the day. Not quite a full day, but I know that this is a Friday afternoon. And so the 4 of us, and actually, David, if you don't mind joining us up here as well. We're going to summarize what was discussed in the breakout sessions, what we saw as potential pathways or roadblocks that we saw, different, additional conversations that were sparked by the prompts in each room. And we will cover across the 4 different breakout sessions. And I believe we're starting with the patient stratification. So Klaus, if you'd like to lead off.
We had a small group, but the conversation was very stimulating. We leveraged Tina's handout heavily, which I think summarizes really well the key points that we discussed throughout the 2 days. We started with proposing a more specific application for an envisioned CIVM for patient stratification, thanks to the input that we got from our pharma colleagues in the room. And so the proposed, and by the way, even if this ends up being diagnosed as not necessarily feasible at this point, the importance and relevance of the exercise was that we were able to walk through the steps to propose an organized context of use. So we started with the notion of leveraging the CIVM for trial enrichment to minimize the inclusion of individuals with a high risk of drug-induced autoimmune hepatitis, based on a very specific industrial need for drug development that was voiced in the group. Does it mean that it's the most important unanswered question right now for drug development? It may not be, but it was a very relevant one anchored on a very specific need of industry that was voiced in the room. So then we went through the handout and the questions, and so we started discussing the key question. And so the key question, which is an aspect of framing a question that goes higher than a specific model or solution, was defined as which population should be selected for clinical trials to minimize autoimmune drug-induced hepatitis. So then moving on to the other pieces of information that could be leveraged to answer that specific question, we understood that it's important to really frame the question and the context of use against the target profile of the drugs being considered. We didn't get into a lot of detail about the target profile for compounds that are proprietary, but we identified that that's important to keep in mind. Pieces of information in the preclinical stage that you can use to inform that decision about trial enrichment when you hit the clinical stage. Animal models, it was pretty much agreed by the group that they're non-existent for such an application. Other types of preclinical bench tools was also determined that they were non-existent, but if anybody knows about a tool that exists, we're of course welcome to get that information. But what was recognized was that information from previous drugs that have gone through the preclinical and clinical stage could be leveraged for understanding what are the factors that drive the risk of autoimmune drug-induced hepatitis. And then based on that, the exercise would have to be on how does an envisioned CIVM fill the gap to better understand that risk. A key driver for that is to understand what is the level of uncertainty that could be reduced that would be acceptable for such a decision-making support in drug development. We talked throughout the 2 days about what if a model gets it wrong. It's not a binary that it gets it completely correct or completely wrong. It's a continuum, and it hinges on understanding by how much the model reduces unexplained uncertainty. And we moved on to the next question of the scoping of the model. This is a model that would be of high scope because certainly no other platforms seem to exist. And of course, a key component of the scoping is that such autoimmune pathways would need to be incorporated. But in order to move to the next aspect of validation and verification, we of course discussed the starting of a fit for purpose verification, but because of the high scope of the potential application, some form of clinical validation would still be required, in which the delta of additional information that the model can provide needs to be compared against clinical trial results. So at the end of the discussion, yes, we recognize that this would be potentially not the most feasible thing to aim for at this moment, but the important outcome of the conversation is that we walk through the steps of defining each one of the questions, each one of the components, to then define a context of use statement. I'll conclude with that. If any one of my fellow compatriots in that work group wants to add anything, please go ahead. Okay. Cool. So that was that.
So this one next to that, Katrina.
All right, and David will help out with this one because he really led the conversation here, along with Jonathan, Daniel, everybody was pretty active in our discussion. So this was the human CIVM model to compare against approved therapies to de-risk. So a lot of good discussion, and I think we didn't really nail down exact details of each of the things in the handout, but we utilized the handout to go through the steps of what we need to think about along the way. So our context of use we decided on and kind of narrowed down to is a CIVM model to prove lower daily toxicity than an approved drug of the same class, and really having a model being able to replicate that known toxicity of that parent compound and duplicate the results. So if you're able to duplicate the results, then that can become a condition of use, and then run the compound of interest that we're looking to test, and other compounds, number of which we didn't come to an exact number of, because as you're going through it, it depends on a lot of different factors. So being able to compare against that known toxicity and the other compounds that you run to really show that relative risk. And then defining the questions of interest and questions coming from the different groups. So from the regulators after you submit your qualification submission, with everything, their question is, do we approve the drug based on everything that was submitted and what we know, and how do we label it? And then when you're going through the qualification, should, at different steps along the way, should we proceed to clinical studies based on the known toxicity that the model is showing, that sort of thing. And then defining the scope of the model, defining all of the attributes of the model, cell types, biomarkers, endpoints that are being looked at and assessed. And then in terms of validation and verification, that model validation, the number of test compounds depends on what specifically you're looking at, and having a really defined context of use and that question of interest. So going back to the details of that. And then the assay validation, wanting to match your dose and your exposure to what you'd be seeing clinically. So those are kind of like the high level details that we got through, but David, please add on what you think.
Yeah, so I just see that from here. Yeah, I think we had a very lively discussion, and we had, I think, the largest group. So probably everybody took away different things from the discussion, and we kept using the worksheet to sort of that Tina made to reorient us, but maybe we kind of let the discussion go where it went, and maybe we weren't so structured. But so I'll give some thoughts on my thoughts on some of the aspects. I'm going to just sort of go through it in the order that was on the worksheet. So which starts with the question of interest, and we were kind of using the question of interest of, you have a drug that that there was a predecessor molecule from the same class that had caused drug-induced liver injury, and it hadn't been approved, but had been dropped from development, like in phase 3 clinical trials, because there were liver injury signals. And so now you have, and I think, like, for what's up here, though, we sort of in our discussion talk through the concepts a lot of question of interest and context of use, and we sort of, I think, went beyond our original assignment, and it was more about the understanding the concepts. So it perhaps got a little confusing to sort of orient back to our. We may have strayed from our assignment, is what I'm realizing. So, but that's okay. So I think the question of interest in this case was, like this example, it's actually a real example, and that is in the public domain to an extent. And it was a example that we covered in one slide into this FDA Science Board presentation about a year ago that I had actually referenced in my talk yesterday. And there are links there to public reviews. It doesn't have great detail, but it has some information. And the context, so the question of interest is, there's now a new drug, another drug coming along in development. And I think some of the we have multiple things in the question of interest here, because we were talking about the question of interest could be different at different stages of development. So, there may have been a question of interest at the time of moving this other molecule into phase 3 trials, because it had a similar molecule, it had a risk. Well, would you have more patient monitoring? And then at the time of approval, how are you going to label the drug? Does it get a warning for liver injury because a similar molecule had liver injury or not? So those could be like questions of interest. And then the context of use, would depend, would ultimately need to be very specific, tying back to the question of interest. And I think we kind of went around and discussed different questions of interest, so it got a little bit, I think, perhaps confusing in our session. But we kind of talked through a lot of questions of interest and context of use in our discussion. And did you want to say something, Tina?
I did, because I think the question of interest you were talking about sounds like it could be a really useful one, in the sense that there was a drug that had some toxicity in a study. Now we have this new device that could come along. If it could predict the same toxicity, but now be used for this new compound, my question would be, what I think is missing here is, what are the other things that would inform on that new drug? Because it wouldn't you guys just wouldn't rely on the CIVM, because well, then that would be like the scope of that would be really high, so the validation would be really high. So what I don't understand is, in the context of that question, what else would FDA ask for in terms of data? And then you want to compare with all of those data on the table, what's the scope of that model compared to the other data sources?
Yeah, and the group did discuss that some. And at the beginning was talking about other, taking into account the chemical structure and like other characteristics of the molecule, and that there was discussion about other in silico model information and how you'll have clinical data, like, are you getting liver enzyme elevation? So there's all these other pieces of information. And so we discussed that a lot, and how your new complex in vitro model is not going to be a standalone thing in isolation. It's part of the integrated assessment of non-clinical and clinical data, and which is really your second part of your worksheet. And then so what is the specific role of the model to address the question of interest? I think this is sort of could be in line. We were thinking about the talk from this morning on how the Dilly team at FDA thinks about questions, and they're taking all the information together, and the approach is like right now it's an expert opinion really of the team. And so if there's a new complex in vitro model, that it would probably be weighed with all those other pieces of information like Tina was suggesting.
Can I offer something with that discussion? So I think the companies that are developing those in vitro models, for if this is a viable question of interest for FDA and the use of these for that, what I would recommend is you kind of take a step back and say, okay, if my complex in vitro model can predict it with, 50% accuracy, how would it fit into the scope, right? How much would FDA rely on it? If I could get to 75% or 80% or whatever the bars are, have that discussion. That way the industry would know, okay, if you're telling me I can get to 80% or 85%, then that's what I'm going to work on, so that this can be a part of that decision making. But if you can't achieve that, well, where does that put me? I think that's the constructive dialogue that I think industry and FDA could have together, if I may say that.
And Tina, no, I think that's a really relevant point. And the other thing is that the context of use statement needs to be an evolving entity. So as you continue to generate the evidence for the development of these solutions, you will always need to go back to the context of use and continue to adjust it. And if the adjusting is still something that adds value to decision-making, drug development, you can proceed. But if the adjusting ends up in a situation where uncertainty could be too high, let's make sure that we identify what are the blind spots and then provide a roadmap for filling those blind spots to then continue. Exactly.
Yeah, I think building on that concept, in Jeff Siegel in his talk yesterday sort of made reference, and we've talked about it in other contexts. So in the clinical biomarker space, there can be different types of biomarkers, and often when biomarkers are qualified, they'll first be qualified for like an easier context of use, and then it may evolve over time. And so for those that are familiar with clinical biomarkers, so there's different types. You can just have like a predictive biomarker or a prognostic biomarker that may, identify these patients are more likely to, well, in the case of, I don't know, develop liver failure, let's say. But maybe later it gets advanced, and now it's a biomarker that it predicts who would benefit from the drug. And then like the ultimate biomarker, like qualification, often is a surrogate endpoint biomarker. And that's like the hardest one to achieve, where instead of following patients to see if you have a drug that's going to prevent liver failure, I'm not talking about like drug-induced liver failure, just let's say liver failure of any kind, and you want to develop a drug to prevent liver failure. Well, if you could develop run a clinical trial with like a small number of patients and demonstrate, use the biomarker, that's a big win. But that's also a hard thing to qualify. I don't know, Klaus, if you have any perspective on that, but it's this parallel of like starting with the easier to achieve context of use.
I'd like to bring up kind of one, actually, I think very relevant qualification we have in process within PSTC, and that's looking at GLDH as an alternative to ALT or AST. And when we started that qualification, we proposed a context of use where it would be a replacement to ALT due to its specificity to the liver over ALT and AST. And when we started to look at what types of studies were needed to provide that level of evidence, we went back and we refined that context of use to a use case that's very needed right now, and that's in the muscular dystrophy space, where these the people with muscular dystrophies have very high levels of ALT and AST as a baseline, and so looking at changes can be very difficult. And so we changed our context of use to when you have a known muscular injury risk with the drug, or in a therapeutic area where there is muscle degeneration.
Yeah, I think that's a really good example of you have this new and I'm sort of familiar with that use case. So you have this new potential biomarker, and maybe it's better ultimately, and you want to replace ALT, let's say. So now let's say you have a new complex in vitro model and you want to replace an animal study. But like in this case, I don't know, the data wasn't there. There wasn't quite the full need or we're just, for whatever historical reasons, not going to replace ALT completely. But there are places, there are areas, types of patients where ALT doesn't work, because if you have muscle injury, your muscles release ALT. So that's like a major problem for drug developers and for regulators, because you now no longer have a way to detect if there's liver injury. So you can actually solve a problem there with a new biomarker. So, maybe the ultimate goal is to replace animal studies, but where are there certain use cases initially where there's a problem with the animal study and you could actually fix it? I think that's a good parallel.
That's a great parallel, because in the biomarker space, and Nick knows this full on, if we had a dime for every time we hear the solution to all our problems is to get a surrogate, we probably would be doing something else. And the conversation in that space has been one in which, from the surrogacy level, we bring it down to understanding exactly what is the added value answer that would address an unmet need. And when you present it in such a way that sponsors go, oh, wait a minute, so actually, without even thinking of a surrogate, I can optimize my patient selection, and that decreases my uncertainty by this much. That helps me with my internal upper management conversations to get them convinced that they should invest in my therapeutic area. That's a big win. That's a huge win.
Yeah.
And there are parallels that could be brought from that space to this space.
Yeah.
That summarizes it. the next one. Retrospective toxicity analysis.
You'll have to excuse, I wasn't quite as neat on the summary here. Came back from lunch a little bit, well, I guess not as late as Klaus, but but our group, our topic was retrospective toxicity analysis. And so this would be a case where you didn't see in any of the non-clinical studies an indication that there would be hepatotoxicity, but you go into your phase 1, phase 2 trials in healthy volunteers, and all of a sudden you start to see liver enzyme increases. And so this is what we put as our question of interest, is that, that you observe liver enzyme increases in several patients. It doesn't lead to a clinical hold, but you see that there's a potential issue right now in phase 1, 2 trials. And you can see a little bit of our thought process is here of complexity that we're considering. So the question here is, does the therapy cause liver injury? And then moving forward with a model, and we also considered, well, if yes, what is the mechanism of injury? And when we started to discuss where the mechanism of injury in terms of assessing that component, it led to many more questions and different types of models that would be needed to address that. And even looking at just liver, if we're just saying liver injury in general, it still is broad in terms of what Eric brought up yesterday, and I'm sure everyone knows. So there's several different types of liver injury that can occur. And so when we started to put together a complex or a context of use, we said that it would be a CIVM that can detect if the injury is due to direct hepatic injury, such as necrosis, apoptosis, etc. And as well, within the patients who showed an increase in liver enzymes, we want to understand beforehand by probing those patients to see if there were any underlying susceptibilities that were not identified before the trial. And then if that's the case, then that would need to be included within any model that's moved forward as the test model, and then as well having a control set of cells that are negative to that susceptibility. So if there's a genetic mutation that leads to a higher DILI risk, or differences in HLA, which has been brought up several times. And then the other questions that come up is which dose levels led to increases, as well as where were the exposure levels, and how would that then feed back into, as we're kind of going through some of the analytical considerations. And so as we're going through this, I think when I thought about this one earlier on, I thought it kind of fell under a step up from what we saw as probably the simplest context of use. But there's so many different directions that this could go in or needs to be assessed, that it becomes a much more complex question to look at the assessment that needs to be carried out. And so that's where we narrowed this down to just direct hepatic injury. And so those are overall kind of the components that we considered. The next piece within in terms of putting together a qualification project would be we already talked about the appropriate cells depending upon susceptibility to types of injury, and including those. And then as well, if we want to start diving into different types of injury, we would need to be having output of measuring different biomarkers that are indicators of those specific types of injury. And so I'm not sure if anyone else from the group has comments you'd like to bring up that we discussed during the session. Yeah.
I have a question, only because there's so much stuff I don't know. David, is this the kind of discussion we were having with the group inside FD? We were talking about the liver MPS chip specifically as a possible, like a potential, like there's a signal, either in a phase 1 study that maybe we could use the chip to like determine if we either pause the study or change the population or do something else? Is that the same kind of thing?
Yeah, I mean,
Yeah.
yeah, I guess
Yeah.
this could be interpreted the same as what was our other, the group we were in some ways. I mean, yeah, I think we talked about a lot of context of use in our discussion, so we sort of talked about this one
My reason for
too.
asking is what information do you use now to decide if, this injury is something you need to stop the trial or change directions? And
I don't know,
how
Eric,
would-
you want to
right?
comment for, as, I don't think some of our hepatologist experts from FDA are in the room still, but no, but yeah. No, but it might help to reinforce, from the sponsor side, you're really thinking about the same questions. What are you, as what FDA would say. So if you have a potential DILI drug, a drug that may be causing DILI that in your company, what are the pieces of data? It's kind of what Skip was covering this morning that you're looking at. In how you think about it from the industry side.
It's complex for sure. Depending on the disease population, patient population being studied, there are different appetites for risk benefit of drug-induced liver injury, the phenotypes that are seen, the severity of the outcomes that are seen, the frequency, the level of evidence for DILI, because it's not always so definite, and the phase of study that it's in. So there's not a good one-size-fits-all answer to that.
Yeah, but I think that's the important part of the thing that I think, yeah, you need, and I think Tina's point on the workshop of on the worksheet, her second bullet is she had identified the different models that will inform the question of interest, but it's not just models or, like patient characteristics, all these other
really
factors.
just like evidence, right?
Yeah,
Data sources, right? Yeah.
If we have a new tool, a complex in vitro model, and if it's being used during decision-making, during clinical stages of drug development, I mean, there's a lot of other pieces of information you're considering, or if it's in
Because the idea of like using one of these chips to decide whether you keep going or you stop your study sounds like a really important context of use for that device, and probably
Right.
would be like the evidence that FDA would want to see would be really high. But that's how you would have that conversation, is in the context of the other information that you would make this decision. You can't put the requirements on that device all by itself. It needs to be, from my perspective, in light of all the other things that you would use to inform that decision. So I was just curious about what that looks like for this case. So that's helpful.
Yeah, I mean, I think that it would involve, I think at least several different types of biomarkers to assess different injuries coming out of it, as well as looking at, obviously hepatocyte death. And so those would be initial components, definitely.
But I think you have to ask, okay, so if you're trying to help someone in Eric's shoes at a pharmaceutical company make a decision, or you're trying to help a review team at FDA make a decision, I mean, you have to know how they're thinking about the problem. And so it's sort of the bigger picture point is to understand who your customer is. If you're giving them a new tool, how might they, try and solve a problem for them.
And I'll add one more detail, and that is within development, it's not just a decision to continue a program or discontinue a program. There's also the nuance of, we now recognize that there is a liver risk. How will this factor into the drug's label in terms of the language that goes in, how strong the language is, what precise words make up that risk language in a label? And so there's all of that detail that can be helped out with
Yeah.
a model like this.
Yeah,
And
and it's kind of to add on to that, potentially with a model like this, it could lead to what the monitoring strategy should be by indicating which, if there's specific biomarkers that are going up and others that aren't, and that would also, lead to the labeling.
And one more comment, Nick, you did a nice job summarizing our conversation. And I want to add one more detail, and that is whether this eventual in vitro platform that we talked about would be something that's just standard, or to what degree will we be able to personalize it, to look at individuals' risk factors and susceptibilities. And if we want to tailor it to the individual, maybe those individuals who have shown ALT elevations in a clinical trial, how do we go about doing that? What is feasible versus just a pipe dream?
Yeah, no, and Mark Abigan brought up an example around using monocytes to de-differentiate and then differentiate them into hepatocytes and grow an organoid, which I think there's at least one company working on a model similar to that, to answer like kind of the precision medicine level answering that type of question. Thanks. Do you have a question?
Seems like a really interesting, important context of use, actually.
Okay, so we were the last group. So our question of interest, our unmet need, was really framed around something that Nikisa started out with her talk, that there are not good models for immune-mediated DILI for reviewers right now. And I apologize because you can't read any of that on the screen right now, but I will talk through it. So really the question came down to what is available in the short term versus long term for immune models of DILI on a CIVM. And in the short term, we thought that there might be some real potential for innate immune response DILI models that we could leverage. And long term, as these models become more complex, once we start to be able to add in T cells or circulating PBMCs or disease models, we might be able to do an adaptive immune model as well. But short term, for a fairly narrow defined context of use, and I'll just read this because you probably can't see it, is to use these systems as a tool for retrospective analysis of innate immune response DILI observed in clinical trials. And this could rule out potential causes of the immune DILI, and it would just be an addition to the existing battery of in vitro testing and existing non-clinical data available to reviewers. And so I approach this immune-mediated DILI as something that was going to be very broad, very general, used per perspective analysis, but I think with this slightly narrower context of use, this becomes much more realistic and much more achievable in the near term. In this slide right here, we went through what's available currently in blue and what open questions are here in red. So currently we have models that integrate Kupffer cells and stellate cells. We can measure IL-6 gene expression, LPS stimulation, drug stimulation, and look for immune response from the Kupffer cells. The outstanding questions really are all around sources of cells, donor variability, HLA type, durability of these systems, and then how we can look at available compounds in these systems. I mean, Kevin Ford's talk sort of threw a wrench into our plans for having a tool compound list, but we will work through this as a community together. So what can we measure right now with these models? I've got that duplicated, that's cool. But really the discussion was very robust, and I think we agreed on this narrower retrospective analysis as a first step forward to try to integrate these systems into a reviewer's toolkit for looking at immune DILI. And we wish we had Eric in the room with us to guide us from a hepatologist perspective, but that's where we ended up.
I really love the fact that you guys thought about I really like that you guys had the conversation of what can we trust today and what is still open questions, but could be on the table if we could raise the bar for that. Like the fact that just adding a different perspective on that is really important to the piece. So that sounds like you guys had a productive conversation too. I'm so excited for everyone.
And Nikki, Lorna, if anybody wants to add comments, please. Yeah. No? Okay. Then I think that's everything from this session.